Hello dear aspirants, very good evening all of you. Welcome to the GDC Class English and I especially welcome you all for the Pharma Thunderstorm series. In this series, I am going to teach you about very important aspects of plant tissue culture. You know that pharmacognosy has a good weightage in all pharma competitive examination. Considering that one, okay. So I am today going to discuss about one of the important topic called plant tissue culture. Plant tissue culture. Okay. So by the end of this topic also, we are going to have a 10 important MCQs based on the previous year papers as well. Okay. But you need to do one thing, that if you are not subscribed to this channel, do it immediately. Subscribe this channel and click on the bell icon and share to this your friends, those who are preparing for all pharma competitive examinations. Okay. Yes. Without any delay, let us start this today's session. Okay. Yes. As you know that plant tissue culture, I'm going to tell you that. Okay. So do remember one thing. The answer will lies in between the lines. I used to believe one thing, that the answer will lies in between the lines. You can get the entire definition or process from within these lines. Within these lines, okay. Plant tissue culture. Okay. Yes. What do you mean by this? Plant tissue culture. Yes. Yeah, from the plant, from a specified plant, if you are collecting a tissue or a cell and if you are culturing under in vitro conditions, under aseptic and in vitro cultivation to regenerate into a new plant, that will be the plant tissue culture. The name itself will defines about the process. Plant tissue culture from a desired plant. If you are taking a tissue or a cell to culture them under aseptic and in vitro cultivation to regenerate, to obtain a new species, to generate a new plant, that the process is known as plant tissue culture. Within this term or within this image, you can find out the process from a plant. Yes, from the desired plant. If you are collecting a tissue or a cell, if you are collecting a tissue or cell, and if if you transfer that to the a specified nutrient medium under controlled temperature and aseptic conditions and in vitro cultivation, thereby you're going to get the desired species, desired plant species. This phenomenon is known as plant tissue culture. Clear? Yes. Whenever you're going to learn anything new, just you need to question three aspects. Okay, what, why, and how. Okay, what do you mean by plant tissue culture? You have learned now. You have learned just now. Okay, you have understood now. Plant tissue culture, you know that if you are taking a tissue from the plant, okay, if that tissue was regenerated into a new tissue or a new plant under aseptic conditions, that could be a plant tissue culture. Okay, that's the definition of plant tissue culture. And why plant tissue culture is needed? Yes, at the end of this tissue culture, you're going to get the new varieties such as polyploidies, hybrid varieties. How you're getting all these things? Because of the tissue culture technique only. Yes, you are combining the two different plant species. You are taking a tissue and you are adding the changes. You are changing the nutrient composition. Thereby, ultimately, we are going to get the new varieties. That's the reason why we require the plant tissue culture. To get the new plant varieties, to get the desired varieties, we require plant tissue culture technique. Without the plant tissue culture technique, there is no hybridization. Without the plant tissue culture technique, there is no polyploidy. Without the plant tissue culture, there is no mutations, and without the plant tissue culture, there is no new varieties, no new plant varieties. Clear? So that's the reason, in order to create and in order to overcome the problems which are there in the plants, we need the plant tissue culture. Clear? How you are preparing the disease-free plants? How you are preparing? How we are generating the disease-resistant plants? By means of a meristem culture. Meristem culture is a one of the plant tissue culture. Clear? So to overcome some problems which are arising in the plants, we are going to rely on the plant tissue culture. Clear? Yes. So I think you have understood, right? And how the plant tissue culture we are going to see. So you have understood what is plant tissue culture and why plant tissue culture is needed. You have understood, I think. Is it clear? Yes. Thank you. Okay. And now let us see what are the different types of cultures and also what is history. Because every method, whatever it may be, whether it is analytical method or biological method that can be developed by an important personality, there is a key person. Definitely, we should remember, because exam I will ask you about that important personalities. When it comes to the plant tissue culture, 𝙂𝙎𝙎𝙎𝙄𝙊𝙉 is known to be the father of plant tissue culture. Okay, 𝙂𝙎𝙎𝙎𝙄. T, it ends with T. T stands for tissue culture, like how we need to remember 𝙂𝙎𝙎𝙎𝙎. Okay, and you can see he is the father of plant tissue culture because why he become and why he is known to be the father of plant tissue culture, because he also, okay, he is the first person, he is the first person to introduce a concept called totipotency. Introduce a concept called totipotency. Okay, again I'm telling you one thing, the answer will lies in between the lines. Okay, within this word you can find out. Okay, toti means total. Okay, total. Potency means ability. Total ability. Okay, total ability to regenerate into a new plant. Okay, the totipotency will be occurred in, will be seen in cells. Okay, so if you want to culture a plant, if you want to culture a tissue or a cell, okay, definitely the tissue or a cell should definitely contain a property called totipotency. If the cell is not containing the totipotency, you are not able to do the tissue culture. Clear? Because that cell has to be grown into a new plant. That ability, okay, that single cell should be grown into a new plant. That ability is known as totipotency. And the person who discovered this is 𝙂𝙎𝙎𝙎𝙎. And another important personality from the history of plant tissue culture is, okay, 𝘂𝙎𝙎𝙎𝙎. Another important personality is 𝘂𝙎𝙎𝙎, and he observed the secondary metabolites in the callus tissue culture. Okay, in the callus cultures. Okay, two important personalities, very very important. The father of plant tissue culture is 𝙂𝙎𝙎𝙎𝙎, and another important person is 𝘂𝙎𝙎𝙎. He observed that secondary metabolites in this callus culture. Okay, and also, yes, if you define this entire process in one line, yes, okay, plant tissue culture is a technique where you are, okay, where you are developing a new plant or where you are regenerating a new plant from a tissue or a cell under aseptic conditions. Sir, why we need to maintain the aseptic conditions? Yes, okay, if you are developing a new plant or a new cell, okay, that should not be interrupted by any of the microorganisms or any other foreign particles. Okay, if they are interrupted, ultimately the growth is going to be interrupted. Ultimately we won't go, no, we are not going to see any type of growth. Clear? Aseptic conditions under controlled temperatures. If you are developing a new plant from a cell or tissue by providing the suitable nutrients, that could be the plant tissue culture. I hope you understand. Yes. Okay, so these are the different techniques, and let us see, okay, in detail each one. Okay, don't worry, and let us see the definition again. Plant tissue culture is a practice of practice used to propagate the plant under sterile condition. That's the reason the nutrient media or the transfer of the explant to the nutrient media will be very sterile in nature. Otherwise the plant will undergo for several mutations and several side effects. Okay, yes, okay. So now we will see the process. Okay, simply we have explained here itself. We can see here. First of all, the principle or the basic process of plant tissue culture will be explained here. First of all, what we need to do, according to the definition, we need to collect the tissue or a cell, and that should be transferred to the nutrient medium. Then only should be grown as a specific plant. Okay, should know some definitions with respect to the plant tissue culture. Thereby you will understand. Okay, so we'll discuss the definition as well. First of all, okay, in order to carry out the plant tissue culture, what we need to do is you need to select a plant where, you know, and which plant you are going to do the tissue culture. Okay, and then you need to select a specified organ. So here we have selected the leaf. Okay, and then we have collected the tissue sample. And this tissue sample which is collected from the leaf, it is known as explant. Okay, from the explant only we are going to get the new plant. Clear? So from the desired plant, if you are collecting a tissue to regenerate a new plant through the callus, that could be the explant. Okay, so simply if you are taking a desired plant part, okay, desired tissue from a plant, okay, that could be explant. Okay, and yes, and after that when you transfer this explant, okay, when you collected this leaf, and from that leaf you have collected a tissue sample, and if you transport that to a nutrient media, because cell are the vital things or viable things, right, it should be grown. So in order to be carry out, okay, in order to be in a living nature, what we need to do, we need to supply the nutrient media. For the reason here, whatever the nutrient is required for the growth, right, micronutrient, macronutrients, okay, pH, amino acids, enzymes, sugars, Okay, for carbon source, all the things are prepared as a nutrient media, and in this nutrient media, after specific time period, it can be developed into a undifferentiated mass of cells. Do remember, definition of the callus is undifferentiated callus or undifferentiated mass of cells. Why it is going to be undifferentiated or why it is grown? Because here you have taken one cell. Okay, so we are providing the nutrient media. So by utilizing the nutrient media, the cell will undergo for several changes, cell differentiation, right? Okay, and cell division is going to happen. So thereby cells are multiplied and undifferentiated mass is going to be formed here itself. Okay, and then again this callus is going to be transferred, and into the suspension culture, and thereby the cell suspension. Okay, we have transferred the tissue into suspension, and thereby we have divide them into a single cells by the suspension culture due to the hesitation reaction. Okay, yes, we have collected the different cells, and these cells again further cultured into a new plants. Clear? Simple. From the plant we need to cultivate tissue. That tissue should be transferred to the nutrient media under aseptic conditions. After aseptic conditions, after transferring the aseptic conditions into the nutrient media or culture media, that particular cell or explant will be regenerated into a callus. That callus can be transferred into a suspension. Okay, after transferring them into suspension, we need to subject them into agitation reaction. When we subject them into agitation, the cells are divided into single cells, and thereby this single cell will be regenerated and redeveloped into the new seedlings. So this was the process of plant tissue culture. Clear? And why the plant tissue culture is needed? These are the applications, very important. In order to produce a virus-free plant, we require plant tissue culture, meristem culture, and also maintain the quality. To maintain the quality only we require plant tissue culture. Yes, if the plant species were eradicated, if any plant species were going to be extincted, we are going to lose the quality and particular quantity or maybe the particular species. So in order to preserve them, what we need to do? We need to preserve their tissues. Cryopreservation. Are you getting my point? Do you know the cryopreservation? The storing of the particular tissues under liquid nitrogen at 192 degrees centigrade. Clear? Yes. So to maintain the species and also quality, soma clonal variation. So different varieties of the plants can be produced by the tissue culture only. Yes, we are we are combining two different species, two embryos, two different cells. So thereby they interchange their qualities, characteristics, thereby they produce a new varieties, that is a soma clonal variation. Micro propagation, you know that's very well. And also genetic modifications. Yes, we can do the genetic modifications because this tissue culture will be starting from the cellular level. That's the reason you can do the genetic modification. And lastly, to understand the biogenesis of secondary metabolites, to understand the biogenesis of secondary metabolites. Clear? So these are the applications. Not only this, nowadays the applications or the use of this plant tissue culture technique was increased tremendously. Yes, so you need to look at some, you need to understand some definitions, but before that, same again I have explained the process in this simple steps. So plant selection, and thereby from the explant collection, the explant is transferred to the next nutrient media. Callus development to plant development, plantlet to new plant. Clear? And now, so first of all, for any competitive examination from plant tissue cultures, they are going to ask some important terms and the definitions. Focusing on that, I have prepared this content. Please go through that. First of all, plant tissue culture. We know that what even by plant tissue culture, in vitro growth of plant parts in an artificial medium under sterile conditions. And what is use? What is use? To produce a mass multiplication research, germplasm conservation. Yes, there are several applications. And next, what do you mean by totipotency? Toti means total, potency means ability. Total ability to regenerate, ability of a single cell to develop into whole plant. Clear? The total ability of a single cell to develop into a whole plant. That is the totipotency. Clear? And yes, it is a basis for tissue culture. It is a base for tissue culture. And coming to that, what do you mean by callus? I have already discussed. Callus means undifferentiated mass of cells which is generated from the explant when we transfer them into nutrient media. You can see here, unorganized mass of undifferentiated cells from the medium with hormones. when you transfer the plant tissue, when you transfer the explant to the nutrient media, that can be developed into the callus. That is a undifferentiated mass of cells. Okay, clear? And yes, it is mainly for callus is mainly used for the organogenesis and somatic embryogenesis. Clear? It is a base. Actually, the callus is also one of the base for generation of the new tissue. Okay, whatever the explant you have taken, ultimately you should undergo for the callus. Okay, explant that should be grown as a callus. From the callus, okay, the plantlet is going to be developed, and thereby ultimately, okay, we are going to get the new plant. Next, explant. Yes, what do you mean by explant? I already discussed. What are the plant bud that you have taken for the culturing? What is the plant bud you have taken for the culturing? That could be explant. Okay, if you're taking a leaf cell, that is explant. If you're taking the root cell, that is explant. If you're taking the roots tissue, whatever the plant, what is the initial cell or tissue you have taken for the culture, that could be the explant. Okay, and yes, that's the source material for culture. Okay, and next, what do you mean by organogenesis? I used to believe one line every time, organogenesis. Okay, organogenesis. Okay, organ means a specific part, genesis means, okay, creation. Okay, within the organ, if you're creating a new plant variety or new seedling or new planted, that could be organogenesis. You can see formation of shoots or roots from the, okay, roots from callus or explant. Okay, if you're directly, if you're directly creating the explants, sorry, if you're directly creating the shoots or roots, okay, let us consider, okay, let us consider from the callus, from the callus or any tissue, if you're directly creating the shoots or roots, that could be the organogenesis. Okay, simple. If you are creating the organ, yes, obviously, let me tell you very easily. Okay, organogenesis, which means you are creating, you are developing organ. Organogenesis, genesis means development. Organ means specific organ. Okay, here, what is organ here? Shoot or root. Shoot or root is an organ. Okay, you are creating or developing on the explant. Yes, clear? If you are developing a shoot or root from the explant, that could be organogenesis. Okay, and next, somatic embryogenesis. Okay, somatic embryogenesis. First of all, in order to understand somatic embryogenesis, what you need to do? You need to know that what is somatic cells apart from the sperm and egg cells. Okay, normal cells. Okay, any cell apart from the, okay, apart from the zygotes, instead of zygotes. Okay, you can create, okay, you can okay, you can develop any tissue from the somatic cells. Okay, if you are using somatic cells, if you are developing the, okay, if you are developing the embryo from the somatic cells, that could be somatic embryogenesis. The name will tell you, somatic embryogenesis. Somatic embryogenesis. Okay, from the, okay, from the somatal cells, okay, from the, okay, somatic cells, if you are creating embryos, if you're creating embryos, that could be somatic embryogenesis. Okay, is it clear? From the somatic cells, if you are developing embryo, that could be somatic embryogenesis. Okay. And next, micropropagation. Micropropagation means tiny. Okay. Micro means smaller. A rapid multiplication of plant using tissue culture techniques. Rapid using, okay, rapid multiplication of plants using tissue culture techniques. And what is the use? A large scale production. Yes, by if you take a mic, see, after developing into a plantlets, okay, and then if you collected them and if you propagate them in a specific area, that could be a micro propagation. Okay, and yes, what is the application of somatic embryogenesis? To produce artificial seeds. Why? Because seeds, in the seeds only we are going to have a embryo. If you are developing embryo, indirectly you are developing seeds. Is it clear? If you are developing embryo, indirectly you are developing the seeds, because the seeds are present in the embryo. Sorry, embryos are present in the seeds. Somatic embryogenesis. Using somatal cells, if you are developing the embryo, that is somatic embryogenesis. Why we need to create the somatic embryogenesis? Because we are creating an artificial embryo, which means we are creating artificial seeds. That's the purpose. Okay? Yes. And what are the other terminologies? Suspension culture. Okay, yes, cells aggregates grown in the liquid medium with agitation. Yes, after developing the callus, that could be transferred into the, that could transfer into the suspension to develop into the new cells. Okay, so mainly for the for production of secondary metabolites and uniform growth. Okay, suspension culture is mainly for to produce a uniform growth, dear aspirants. Okay, please remember. I think now it's visible, right? So no need to worry. I can understand your concern. Okay, next, protoplast. What do you mean by protoplast? Okay, protoplast. Protoplast. Protoplast is nothing but a naked cell or cell without a cell wall. Cell without a cell wall. Why protoplast? Okay. So why we are using protoplast? Yes. If a cell is not having a cell wall is nothing but a barrier. Okay. If you're not having cell wall, the cell can generate, the cell can grow into any direction or any manner. That's the reason if you're using protoplast. Protoplast means, okay, absence of the cell wall. If a cell is present without any cell wall, that could be a protoplast. Okay. Yes. So protoplast fusion and genetic engineering. Okay. To develop the protoplast fusion and genetic engineering, these protoplasts are helpful. Yeah. Mostly this is also one type of technique, which means this is one type of base material. By using protoplasts also, we can develop new plant varieties, because simply you are removing the cell wall. Okay. If you are removing the cell wall, the cell will be unstoppable. Ultimately, it can grow it can invent the changes whatever it needed or whatever it is going to be exposed. Okay. Yes. Next is anther culture. Okay. Anthers are the male reproductive organs of the flowers of the plants. Clear? That's the reason it's known as androgenesis. It's known as androgenesis. Clear? Yes. Simple. No need to worry. Only one thing. If the plants, the term itself will tell you plant tissue culture. From any part of the plant, from any part of the tissue, if you are culturing, that could be a plant tissue culture. So that whatever the tissue you have taken, okay, if the whatever the tissue you have taken, if you have started the culture from the particular tissue, the name will also comes from the like that. Okay, simple. If you are using anther culture. If you are using protoplast culture. If you are using root culture. If you are using shoot culture, because shoot cell we are using, that's the reason shoot culture. Okay, a root we are using root tip culture. Okay, like how. And next, okay, culture of anthers and pollen to produce a haploid, and mainly anther culture is mainly to produce a, okay, haploid or double haploid plants, which means, okay, anther culture is mainly to for the polyploidy. Okay, polyploidy, it can induce the polyploidy. Clear? Anther culture. Clear? But what we need to add in this anther culture in generally, if we add a colchicum atom nil was a natural polyploidy inducing agent. Thereby, we can get a more number of polyploidy agents. Okay, polyploidy plant species. Okay, but do remember only one thing. Anther culture means A stand for anther, A stand for androgenesis. Okay, clear? It is going to produce a haploid and double haploid plants. And next, soma clonal variation. Next is soma clonal variation. Okay, so which means what do you mean a soma clonal variation? Soma clonal variation. Okay, from same genetic variation among the plants regenerate from the tissue culture. Okay, yes, if you have developed a plant tissue culture, from that if you have developed 2 to 3 plants, okay, from the same tissue culture, if you are getting the 2 to 3 plants, but if these are different from each other based on their characteristics, that is a soma clonal variation. Because from soma means same cells, from the same cell we have cultured, and also we got a 2 to 3 different plants with the 2 different characteristics, that could be soma clonal variation. Okay, soma clonal, same cloning but variation in between the plant species. Okay, soma clonal variation. Okay, so for plant breeding and new triads it could be helpful. Okay, and yes, next, cryopreservation. Okay, cryopreservation means just simply storage of cells or tissues in a liquid nitrogen under 190 degrees centigrade. Under 190 degrees centigrade, it is mainly for the long-term germplasm conservation. Okay, long-term germplasm conservation. So most of the seeds or most of the tissues, okay, in the seed banks, in generally they use this method cryopreservation. Okay, under 196 degrees centigrade, minus 190 degrees centigrade, minus 180 degrees centigrade in a liquid nitrogen medium. Next, artificial seeds. Simply, okay, if you encapsulate the, what do you mean by seed? Okay, it's an embryo mainly. It is a development or a mature part of embryo, right? Simply, artificial seed means artificial embryo, encapsulated somatic embryo or shoot buds that acts like seeds. Okay, example, mainly to easy storage and for transport the large scale of propagation, usually artificial seeds are going to be produced. Okay, and next is embryo rescue. So in generally in vitro culture of immature embryos from the interspecific and interspecific crosses. Simple, embryo rescue to preserve that. Okay, to preserve that embryos. What we need to do here? In vitro culture of immature embryos. Okay, just in vitro directly if you are culturing the embryo, okay, in between two interspecific crosses and interspecific species, that is embryo rescue. And lastly, acclimatization. The name will telling you about that acclimatization means, okay, acclimatization, climate here itself, climate, okay, which means to adapt the climatic changes by the plant, okay, and which means we are just hardening the plants or the tissue, okay, to adapt the, see, if the plant was or the tissue or the callus was gradually adapting the changes that are happening in the environment, that is nothing but a acclimatization. Just simply acclimatization, it is, no, it is acquiring the changes, adapting the changes, that is acclim, okay. So mainly to survive in the greenhouse conditions, okay. So these are the important terminologies, okay. And now again I'm going to show you about regeneration techniques. Yes, we have seen the important terminologies. From that definitely you're going to get the questions, okay, clear. That's are those are the important terminologies in terms of any examination, okay. Yes, next, regeneration technique in the tissue culture. Yes, in generally the plant will be from the from the tissue, okay, ultimately the callus is going to be developed, the callus is going to be regenerated, the planted is going to be regenerated by means of which process? We have seen the entire steps, but the plant was regenerated, or the from the explant the plant is going to be regenerated. But for this regeneration process we have these methods, okay. For the regeneration techniques are nothing but organogenesis, okay. We have seen organogenesis, okay. If you're directly developing, any organ from the specific part, okay, so that could be organogenesis. You can see here formation of shoots or roots directly from the explant, okay, directly from the explant, clear, or indirect directly from the callus. Simple. Organogenesis, within the word you can break it. Organogenesis, you are developing an organ from a specific part, okay. If you are directly developing the organ like shoot or root from explant, that is direct organogenesis, because ultimately explant is the first step, first part, okay. Next, if you are developing the shoot or root from the callus, that is indirect, clear. If you are directly creating or developing the root or shoot from the explant, that is organogenesis, direct organogenesis, because direct part, first part we are taking is explant, okay. Indirect means after explant only we are going to callus. That's the reason callus becomes an indirect organogenesis. Examples are like applications. Obviously, micropropagation, colonal propagation, virus free plants. Okay. And next, we know that already somatic embryogenesis. We have seen this terminology already before. Okay. Somatic embryogenesis. Development of somatic cells from the embryos. From the embryos, not zygotes. Okay. Not any eggs or maybe sperm cells, okay, but simply we are creating a new cells apart from the zygotes from the embryos. If you are developing the somatic cells from the embryos, that is somatic embryogenesis, okay. Somatic embryogenesis from the somatal cells, if you are creating the embryo, that is somatic embryogenesis, okay. Somatic embryogenesis, okay. And next, mainly for what? Embryo means ultimately embryo is present in seeds. That's how we need to co-link. That's how we need to interlink, okay. Artificial seeds, large scale product propagation, genetic transformation studies, okay. Yes, these are the somatic embryogenesis. Next, protoplast regeneration. There's another method. By protoplast regeneration also the plant is going to be regenerated, clear, okay. So obviously if you are remove the cell wall, ultimately if you are supplying the whatever the needs, nutrients, water source, carbon, sugars, everything, okay, ultimately from that naked cell, from that cell, because it contains a protoplasm, right, and from that protoplasm also new cell is going to be regenerated. That's the reason we can consider the protoplast regeneration as one of the regeneration technique in the plant tissue culture, okay. Yes. So if you are using a naked protoplast to regenerate a new plant, ultimately that is a protoplast regeneration technique. Mainly applications are somatic embryogenesis, okay, hybrid formation and genetic engineering. And next, ultimately another technique. Okay, another technique is that haploid or double haploid production. Haploid or double haploid production, which means haploid means we already discussed. Haploid or polyploidy means ultimately anther culture. Okay, so anther or pollen culture. So plant produced from the pollen or anther culture, that is haploid. Okay, then chromosomes doubling is haploid. Clear? Simple haploid or double haploid production is also one technique. Clear? Simply by anther and pollen culture also we can regenerate a new plant. Please, dear aspirants, do read this or do understand this in a very practical manner. Apply this. Okay. Try to understand the concept. Don't rely on the words or terms. Just think of it. Whatever explain, just go through it again and think of it. Thereby you will build the concept. Okay, if you build the concept, you cannot forget the things. If you buy hard, you won't get anything. Okay, clear? Yes. So obviously haploid or double haploid production ultimately used for plant breeding, development of pure homozygous lines. And lastly, somatic hybridation, protoplast fusion. Somatic hybridation, protoplastic fusion, which means obviously somatic hybridation means simply fusion of two different protoplasts. If you are simply combining or fusing two different protoplasms, that is a somatic hybridation. It is mainly to create a new somatic hybrids. To create a new somatic hybrids, and it's also known as protoplast fusion. Protoplast fusion. Clear? So these are the regeneration techniques. But ultimately, we are going to see the another important thing called what are the types of tissue culture techniques. What are the types of tissue culture techniques? These are different types. Okay, simple. Nothing to nothing to feel like a difficult. Simple way, because callus culture. Simply, if you are using callus as a base material, as a starting material, as an explant, that is callus culture. Simply, you know the definition. What do you mean by callus? Undifferentiated mass of cells. Okay, mainly it is going to produce the explants. Okay, from where source? From which source we are going to get? From the leaves, from the stems. Okay, we are going to get the callus. Okay, ultimately, mainly, what's the purpose of this one? For to produce secondary metabolites and regeneration and genetic studies. You know that. Next, suspension culture. Okay, suspension culture. Simple. The cells aggregates grown in liquid medium with shaking after developing the callus. Okay, this callus is going to be transferred into the a specific suitable suspension medium. Thereby, the plant will be grown. Okay, in a liquid media, but mainly to attain the uniform growth. Okay, mainly to attain the uniform growth as well as large scale production. Large scale production. Okay, but they are derived from the callus only. Yes, first from the first callus is formed to create a uniform growth, and for the large production, okay, we need to transfer into a suspension medium. Then if you are culturing the callus in suspension medium, that is suspension culture. Okay, and next, protoplast culture. Simple. Okay, ultimately simple. Dear aspirants, do remember only one thing. In the nutrient media, if I place culture, if I place callus, okay, and from the callus if I regenerate a new plant, that is callus culture. Okay, and if I place a protoplast, a naked cell, into this nutrient medium, okay, if I place a naked cell, that is a protoplast here itself, okay, and from this protoplast if I regenerate a new plant, that is a protoplast culture. Okay, if I take an embryo here, if I keep the embryo in the nutrient culture, and if from the embryo if I create or if I develop a new plant, that is embryo culture. Okay, if I take anther or pollen in this nutrient media, okay, and from the anther and pollen if I develop a new plant, that is anther and pollen culture. Okay, and organ culture, somatic embryogenesis, if I develop an embryo by using two somatic cells, that is somatic embryogenesis. Okay, hairy root culture, if I induce, okay, if I use agrobacterium rhizogenous to this nutrient media, as yes, from that leaf or shoot, after adding this rhizogenous bacterium, agrobacterium rhizogenous bacterium, it hairy roots are developed, that is hairy root culture. And from that hairy root cultures, again I further culture them into new plants. Clear? Okay, simple. Callus culture, suspension culture, protoplast means naked cell. If you are using naked cell, that is a protoplast culture technique. Okay, mostly they are prepared by the ice. Okay, two methods are there to prepare this protoplast culture technique. To prepare a protoplast, we have two methods: mechanical method and enzymatic digestion. Okay, simply if you add some enzymes, they partially digest cell wall, then it becomes free from the cell wall. That's the one method. Another method is mechanical method. Okay, if you are simply adding this plant tissue, okay, the leaf tissue in a mortar and vessel, if you simply triturate, thereby the cell wall will break down, thereby that become a cell wall free. Okay, like how there are some methods, and mostly it is mainly for the somatic hybridization. Okay, transgenic and genetic engineering, it's going to be helpful. And embryo culture, okay, in vitro growth of isolated embryo. Simply if you are developing a plant by using embryo, that is obviously embryo culture. Okay, so isolated embryos from seeds and hybrids. Okay, hybrids and embryos from the seeds, we can develop this embryo culture by using, okay, embryos from the seeds, we can culture the embryo culture. Okay, mainly what is the application here? To overcome the seed dormancy, and also embryo rescue in hybridation, to preserve the embryo. Okay, and yes, next, anther and pollen culture. We know that obviously if you are taking an anther or pollen and if you are using them as the explant for culturing, that is anther and pollen culture. Okay, source material is anther and pollen. Okay, mainly to produce the haploids and double haploids. And next, organ culture or organogenesis. Simply, okay, if you can take any shoot or root or leaf, okay, mainly any part of the part like stem or root or anything, that is organoculture. But mainly this should be very very important. Do remember, okay, this is a very familiar technique. Okay, in order to create a virus free plant, this become very familiar. Meristem culture. By using meristem culture only, we are going to get the virus free plants. Okay, colon propagation and metabolic studies also we can achieve. And somatic embryogenesis we have discussed already. Next, hairy root culture. The roots induced by agrobacterium rhizogenes. Simple. If I take any leaf, right? If I take any leaf or shoot or anything, right? So then if I induce, okay, if I take any part of the, if I take any part of the leaf or maybe shoot, okay, and simply if I add agrobacterium rhizogenes, okay, agrobacterium rhizogenes, do remember, this will has the ability to regenerate or to create or to induce the roots, hairy roots. That's the reason this particular microorganism, okay, will induce the hairy roots. That's the reason it is known as hairy root culture. Again, after developing that hairy roots, okay, that hairy roots has been isolated and they are transferred to a new plant, they are transferred to the nutrient medium. Again, from that we are going to develop the new plant species. Okay, yes. So leaf explants infected with the bacteria. Mostly leaf are more suitable for this technique. Okay, so to the leaf, if we add this agrobacterium rhizogenes, they will, okay, induce the hairy roots and thereby they will, okay, develop this one. Yeah, yes. And they mainly for the, what is the significance here itself? Stable metabolite production and secondary metabolites research. That's a main important significance or application. And also last but not least, protoplast fusion. The name itself will tell you protoplast fusion. Two different protoplasms, okay, two different protoplasts. Okay, so obviously two different varieties, two different species, which means, okay, creation of new somatic hybrids, creation of new somatic hybrids. Okay, new somatic hybrids. Okay, and lastly, endosperm culture means the culture of triploid endosperm tissues, mainly to produce a seedless fruits. Very very important. Okay, endosperm culture is mainly to create the seedless fruits. Okay, endosperm. So by taking the endosperm, okay, simple. Whatever the, okay, prefix is there, prefix, the first word is there, that is explained. Simple. In the tissue culture, in the types, whatever the, okay, prefix or the first word is there, that is a ultimately the explant. Here the callus is explant. Okay, either suspension is the main medium, protoplast is explant, embryo is explant here, anther and pollen is explant here. Here we are using organ as explant. Okay, somatic embryogenesis, from the somatic cells we are going to create the embryos. Okay, hairy roots here, the explant here. Okay, and protoplast fusion and endosperm culture. So these are the different techniques in the plant tissue culture, but ultimately we are going to, okay, we need to discuss the ultimate part, and the last part is, okay, yes, ultimately we have discussed, okay, so we have discussed about explant, we have discussed about callus, but the callus was developed in a nutrient media. So do you tell me, sorry, do you know what is the composition of nutrient media? Can anyone comment on the nutrition media? Okay, what is a nutrient media, nutrient composition required to carry out the plant tissue culture? Any one of you please comment. What are the basic sources? Because from the nutrition, from the nutrient media composition also we are going to get a lot of questions. That's the reason nutrient media requirements we are going to see here. Okay, nutrient media requirements. This is a very very important thing. In generally, we know that I think we are more familiar with the, okay, I think we are more familiar with the preparation of culture media in the microbiology, in the biotechnology, right? You are more familiar. Okay, the same media, even though the microorganism is a living cell, the plant cell is also a living cell, right? Yeah. Both are living. That's the reason both do require same nutrition compositions. But let us have a look on the nutrient composition. Okay, firstly, to make it stable, okay, obviously to make it stable, ultimately plantlet should be developed, right? The plantlet should be developed. Okay, every plant is going to be developed and every plant is going to be erect from the soil, because the mechanical support is provided by the soil. That's the reason the plant will be in erect nature. It will be stand towards the sky, right? But the seedling, which is the seedling or the callus is also developing, that also should be grown stably. That's the reason we should add a gel agar mainly for the mechanical support for the growth medium. Okay, and also this agar will facilitate the all nutrients and water. Okay, next, yes, in this next is inorganic salts. In this macronutrients are there, like, okay, sodium, potassium, okay, and also calcium, okay, sulfur and magnesium. These are all the sodium, okay, phosphorus, potassium, calcium, sulfur and magnesium. These are macronutrients which are required more than 0.5 mole per liter. Okay, mainly these two macronutrients and micronutrients are in all comes under inorganic salts. Mainly they are meant for the cells and tissue growth. So to carry out the, okay, osmosis in the plant, to maintain the, the particular, what we can say, the particular composition, liquid composition of the plant, majorly they require this macro and micronutrients. Okay, these are macronutrients and these are micronutrients: ferrous, zinc, okay, manganese, copper and boron and molybdenum. Clear? These are all the things in organic salts. And next is, yes, obviously, in order to carry out the photosynthesis, obviously they need to prepare their own sugars, right? So for the reason, what you need to do for the carbon source, do remember, for the carbon source we need to supply sucrose and glucose. Do the sucrose and glucose contain the carbon? Yes, C6H12O6. In that, 6 carbons are there, right? So glucose contains C6H12O6. Okay, which means here the carbon source is coming from the glucose in form of 6 carbons are there, right? So do remember, for carbon source, sucrose and glucose we are going to supply. Okay, 2 to 4%. Do remember the concentration. Mainly, what is the purpose of this one? For energy and essential ingredients in the general preparation of the culture media. Okay, and pH is very very important. pH is around 5.5 to 5.7. Okay, slightly acidic, clear? Okay, maintain the acidic and basic in nature, both. And important vitamin is B1 thiamine, very very important, essential, very very essential vitamin is thiamine, nicotinic acid and pyridoxine, mainly to improve the cell growth itself. Okay? And yes, important amino acid is glycine, very very important. Do remember, very very important. It can stimulate the growth. Okay, and also they require a plant growth regulates the plant hormones such as auxin. Okay, auxin facilitate the cell division, root differentiation, and induction of growth. Obviously, normal functions. Okay, but plant do require auxins. Okay, and cytokinins for the, okay, cell growth. Obviously, but the gibberellins also important for the enhance of callus growth. But whereas abscisic acid is not for the plant growth. Okay, it is mainly for the, okay, induction of embryogenesis. Do remember this one. Okay, mainly it is for the induction of embryogenesis. Clear? And last but not least, temperature is 10 degrees centigrade. 10 degrees centigrade for the best growth. Okay, so this is for today's plant tissue culture, dear aspirants. Okay, whatever the examination, definitely the question should be coming from this components only or from this concepts only. Okay, you need to understand what is the plant tissue culture, the important personalities. Okay, and also what are the, okay, uh types of techniques, tissue culture techniques, what are the regeneration techniques, and what are the nutrition components or nutrition requirements. Okay, I hope you're ready. Now I think you're ready. Okay, I think you're ready for the 10 important MCQs which have prepared from the previous year examinations. Okay, yes, but do not forget to mention how many right answers you have got from this question. Okay, yes, I hope you're ready. First question of this uh, topic, first one. The phenomenon of regenerating the whole plants from isolated protoplast is known as, okay, from the isolated protoplast. What is the right answer? Please do not forget to, okay, mention the right answer. Try to comment down by adding the question number before. Okay, 1, option A or B or like that. Okay, the phenomenon of regenerating the whole plants from isolated protoplast is known as Undoubtedly, okay, totipotency, okay, regenerating a whole plants from a cell or tissue. Okay, very good. Yes, which vitamin is essential for routinely added to the plant tissue culture? Just now we have came across that slide. Which vitamin? Which vitamin it is? Ultimately, it is vitamin C or E or B1 or vitamin K. Yes, thiamine B1 thiamine is essential vitamin for the plant tissue culture. Okay, very good. Do not forget to mention it is in the comment section. The plant regeneration method is involving the shoots and roots arising directly or via calyces. If the plant is directly regenerated from a shoot or okay, direct organogenesis, right? Direct organogenesis, that's what we have discussed. Okay, if the plant is okay, if any shoots or roots are directly arising from the explant or callus, organogenesis, especially direct organogenesis. Okay, yes, please reread the question, thereby you'll understand. The first successful plant tissue culture was carried out by either by you or me. No, not by you or me. From among this one, option A, 𝘖𝙑𝘏, B, 𝘂𝙀𝙎𝙎, or Gamboge or White. No, ultimately, 𝘖𝙑𝘏 is the first person and he is the known to be the father of the plant tissue culture. Okay. Yes. Next, which of the most important vitamin required tissue culture of the plants? Again. Okay. So obviously, thiamine. Okay. These are the questions which have repeated from the last year paper as well. Okay. Two different types. Same question they have asked twice, but in a different manner. Okay. Ultimately, you know that the thiamine. Okay, thiamine is required. Next, one of the following is not required for the initiation and maintenance of the plant tissue culture. Not required. Which of the following is not required to initiation of the plant tissue culture? For initiation of plant tissue culture, which is which one? Which one is not essential? Sucrose is essential, oxygen is essential, kyanine is essential, right? But abscisic acid is mainly for induction of embryogenesis. We have seen in the table, right? Yes. Next question. An essential ingredient in the general preparation of tissue culture. Essential. Okay. What are the essential thing? For us, essential thing is rice, carbohydrate. Okay. Rice is nothing but a sugar, which means ultimately, okay, sugar and glucose. That's the basic, basic source, essential thing we have discussed already. Carbon for carbon source. For energy also, the sucrose and glucose is required. That's the reason that is an essential ingredient in the culture media. Next question. Meristem culture is most effective method for preparation of which of the following? Bacteria free, virus free, fungus free, or none of the above? Meristem, already we have discussed. Okay. Virus free plants are okay prepared, or it is meristem culture is most effective method for the preparation of virus free plants. Okay. That's the right answer. Okay. And next, the pH of the plant tissue culture medium was usually adjusted to option A 4.5 to 5, 5.6 to 5.8, 6.2 to 6.5, and 7.0. What is the right answer? Okay. Ultimately, the right answer is 5.6 to 5.8. Okay, that's the reason. And the last question in this series, I hope you understand everything till now we have discussed. Okay, last question. Hairy root culture is induced by which of the following thing? Agrobacterium tumefaciens, Agrobacterium rhizosinus, Bacillus subtilis, Rhizobium species. I know you're definitely going to answer this one in the right way. Okay, undoubtedly option B is right, Agrobacterium rhizosinus. Okay, so yes, dear aspirants, this is for today's session, today's thunderstorm series. Okay, so from this concepts, from this entire video, okay, you can answer. I can assure you. Okay, I can assure you that you can answer any question if it is comes from this topic in any examination. If you are gone through this video, you can able to answer all the questions which are coming from the plant tissue culture. Yeah, yes. But finally, do not forget to subscribe this channel. Okay, if you are newly watching, and share this to your friends so that it will be helpful for them also. Okay. Yes. Thank you. Have a great day. Jai Hind.